Recording in progress.
Good day, everyone. Welcome back. I hope you enjoyed your break. Does everyone feel refreshed? No? Yes? We're almost there. It's March by this week, so only 1 month left. Okay, seems a little loud. Today, we're gonna do an activity, and it's gonna require both Clicker and CrowdMark. So if you're not already signed into Clicker, go ahead and sign on. Okay, just a reminder, you have a deep question. It's going to be released tonight, 7 p.m. And did you guys notice more and more the days are starting to seem like days and the nights are starting to seem like nights? We're entering back into the regular, rest of the world having a light cycle. The sun's getting up, it's up before, you guys are probably not up that early anyway, so you don't care about that. But anyways, the sun's coming up earlier and earlier. Okay, so deep question released tonight. And Thursday's class, you can come in to work on it, with your groups or whatever. As always, I'm here to answer questions. It's not going to be a Q&A session, but you can still ask me questions. That's why I come. So that's going to be due this Friday. You got a few days to work on that. And remember, please submit it to both CrowdMark and Critic. Okay? I know vast majority of you are doing so, but you get a few emails every once now and again, people that forget. Okay, so I think you're all up to speed on the learning outcomes for this week. I don't really need to go into that. We're going to discuss gene regulation, and the focus of this week is mainly we're talking about the level of transcriptional control, which is up here. And epigenetics. All right, epigenetics is it's very, very important. You'll recall that the DNA in eukaryotic organisms, it's not just DNA. The genome actually has proteins as well. And those proteins help regulate the expression of the DNA and hence the functions of the genome at any particular cell type at any particular time. So very important to understand how epigenetics regulates gene expression. And I guess the major concept is how you can have identical DNAs, genomes in different cell types, but due to differences in epigenetics, there can be completely different expression profiles and different functions being performed in identical cells with identical DNA, right? All right, so that's what we're going to discuss more detail today. Okay, so in groups of 2 or 3, what could stop you from giving this human-sized DNA a hug? Go ahead. No, I'm serious. Talk about it with your peers and just a few minutes. Try to be as specific as you possibly can. Terminology, use correct terminology. This is not a joke, it's a real question. And if you have a spot open beside you and you see somebody just sitting alone, please wave them over. Tell them to come on over and join your group. It's always best to discuss. Okay, so what's the reason? Yeah. The structure of DNA. Okay, so the structure makes it difficult. Yeah. But it's kind of cylindrical. I could probably still get my arms around it, right? It'd be awkward, but just like, anything else? Yo. Okay. Okay, but this is human-sized DNA. Human-sized DNA. So you're saying it's too small, but you're packed with histones. You're on the track. You're on the right track. The DNA is packed with histones. Hugging DNA. What do transcription factors do? What do histones do to DNA? They grab onto it, right? They're going to grab onto it. They're going to hug it. That's what we're talking about here, hugging. So if you tried to hug a DNA, why would you, a lot of the time, you wouldn't be able to do it. Why? Like proteins, like histones. Why? Sometimes the transcription factor or histone can't. Why not? Okay. Eukaryotic DNA is packaged. It's packaged, very complex packaging. It's wrapped around histones, right? And so you got human-sized DNA wrapped around a histone. You can't really get your arms around it. It's already bound to a big protein. How are you going to, and the transcription factors, they feel the same way. They're coming in, they're trying to do a landing. I found my sequence, I'm going to bind it. And then they go to bind it. And if there's a big histone there, histone's like, no, this is mine. Get off of this thing. This is mine. So it's going to block the protein from wrapping around and grabbing the DNA. Does everyone get that? The histones, that's what I'm talking about here. These histones. And if they're bound tightly to the DNA, well, eukaryotic DNA is not naked. Generally speaking, stretches of they're bound to histones. If the histones are bound tightly, which is dictated by the charges on the tails of the histones, by the code, the post-translational modifications that are on the tails. If it's bound tightly, other proteins cannot bind. Can't give the DNA out. Okay, so, and it's not just like how tightly they're bound, because they can be very, very tightly bound, but it's also due to their positioning, right? Because you could be tightly bound, and what if there's one or two genes in between? That exposed DNA could be hugged by a transcription factor. It could be expressed by a polymerase, RNA polymerase. So it's also it's not just how tightly those proteins are grabbing on. That is a big, they have to kind of let go or be loosely associated to let polymerase, the machine, pass by, right? But also it's their spacing. If they're all really very densely associated, then the transcription factors that go in there and bind, they can't get at the sequences. They just can't. Okay, so spacing, location, and how tightly they bind, very important. So when a gene is being expressed, generally the histones are loosely associated and they're spaced further apart. When the gene is turned off or repressed or has lower expression, the histones are closer together. They're gripped more tightly onto that DNA. Okay, so what time is it here? You have an activity to do, and is it available on eClass? That's kind of why I'm checking the time. I think it should be available now. So open the activity worksheet in groups of 2 or 3. One person records the answers, but submits on the entire group's behalf. Make sure you add all your group members' names. Please go ahead and do this activity. Is it available? Yes. Okay, great. I'm going to give you about 15 minutes, and then I'm going to check back in and find out how we're doing. Maximum I'm going to give you is 20 minutes. Okay, go. Set a timer for 20 minutes.
Recording stopped.
Okay, how are we doing? 2 minutes? 5 minutes? 5, come on, 5, all right. Okay, guys, another 1 minute or so. You can always just submit it before the end of the class. That should be fine. Okay, so let's get back to it. You can submit it anytime before the end of the class, should be fine. And if you couldn't get it done, you can always complete the outside of class version, which is due tomorrow before midnight.
Recording in progress.
Okay, so Yeah, so in the activity that you were doing, you had two different lines of melanocytes. Melanocytes are skin and eye cells that produce melanin. So two different kinds of melanocytes. You have the darkly pigmented, the DP, and the lightly pigmented LP. And so they had two different SNPs and two different levels of expression of OCA2. We know our OCA2 gene that's involved in pigmentation, right? All right, so I mean, you want to remember, I just try to remember one of them so that throughout the examples you can have a reference. So the DP, darkly pigmented, it had a high expression of OCA2 and it had the SNP-T allele. All right, okay. So one of the questions you're looking at, the higher OCA2 expression is linked to more pigment. Okay, so the second question was, in your CHIP assay, CHIP, you're doing like a pull down, you fragment all the DNA, and then you use an antibody that's going to pull down a particular protein. So every time you hear CHIP, you have to think, what protein is it that we're pulling down? And you're not measuring how much of the protein gets pulled down, but you're measuring how much of the DNA target. Okay, so what they did, they did a CHIP assay, compare the amount of RNA polymerase bound to the OCA2 promoter. So they're pulling down with RNA polymerase, and they're measuring the sequence, the promoter sequence. Now, so your results shown in the graph to the right, the red bar on the graph represents what? So you never mind the black and the gray bars, those are controls. So you have the one cell type that's showing you this, the other cell types is showing you that. High level of what's on the y-axis, RNA Pol2. So what they're saying is, well, pulling down RNA Pol2, actually they're measuring the how much promoter is there, so how many copies of promoter, and that's going to be equivalent to the amount of RNA Pol2 that got pulled down. So the red bar represents what? The darkly pigmented cells or the lightly pigmented cells? Which one is it? Oh. Got to open the poll, right? Yeah, get the participation points. There we go. All right. It's a few seconds. Five, 4, 3, 2, 1. Okay. 86% of the class says A. 1% of the class says D. All right. Why do you say A? Why do most people say A? You're pulling down RNA Pol2. Okay. Measuring the amount of transcripts, that's going to give us a big bar here. This is reflecting Pol2 levels. Why do you say the darkly pigmented cells have more Pol2 at that promoter? Yeah. Okay. More RNA means more transcription. Okay, so so you're saying that we measured Pol2 levels, right? Pol2 by measuring the amount of promoter sequences that are present, and you're saying that in the dark cells, the pigmented cells, I'm expecting to have more expression of that gene, more transcription, more expression of the product that's going to make the pigment, the dark pigment, more pigmented. So it's going to be more darkly pigmented. Very good. So, yeah. So does everybody get that? So transcription is not just like the RNA pol binds and then one time and then it goes and it makes a transcript and then everything's done and it's going to be pigmented because there was one transcript. It's not like that. Okay, there'll be a little bit of transcripts even in the light cell, the cell that has low levels of expression. But in the dark cells, there's more expression. Pol2, there's iterative rounds of binding. Pol2 in any one given cell, this mass of cells that we harvested the Pol2 from, if I have 1,000 cells, let's say 900 of those cells are going to have Pol2 bound to that promoter because it's being highly expressed in those cells. Okay, so when I pull down the Pol2, so I would measure Pol2 levels. Yep, that's going to be the dark cells that have higher expression should have more Pol2 bound to that promoter. More expression. Very good. Does everyone get that? Please ask questions if not, because I don't want people getting steamrolled whenever we get the deep questions and it's getting too complicated. Stop me now, or at least ask questions after class or next period. Okay, so question number 3. What would happen if you made histones more negatively charged? What's a histone in the picture? Those little ball things, right? Yeah, histone. What happens if you make it more negatively charged? What's going to happen? Okay, 5, 4, 3, 2, 1. For a class that has 300 people, we rarely reach above 200. That's all right, as long as we complete it. Okay, so vast majority, 89% again. Maybe the same 89% as last time. Maybe not. But 89% of the class is saying B, DNA would wrap less tightly around the histone. Okay, all right. The DNA would wrap less tightly around the histone. If the histones were more negatively charged, how did they become more negatively charged, by the way? Just general terms. How do histones become more negatively charged? It's their tails, these things that are sticking out like this, that actually get the charges, positive or negative, put on them. That's the histone code. How do they become more negatively charged? Well, enzymes come along and they transfer chemical modifications to the tails. Covalently make the tails modified so that they have a negative charge. They can also modify to have a positive charge as well, right? Okay, so whenever you make the tails more negatively charged, they're going to wrap less tightly around the DNA. Why? What charges the DNA for the most part? The backbone of the DNA. You hear negative, 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 negative. The backbone of the DNA has a negative charge. Okay, so opposites repel. No, opposites attract. Same charges repel. So if those tails are negatively charged, they're not going to want anything to do with touching the DNA's backbone. If they're positively charged, though, they grab on, and they don't let go until something comes along and modifies them again. Right, so the DNA backbone is negatively charged, and so histone acetylation, remember that modification, acetylation increases the negative charge of the histone. It's particularly of the tails, like we were talking about, and then the histone would then bind the DNA less tightly. And so if you want to have a gene expressed, you want to have access to that DNA by the RNA polymerase or by the transcription factors, what needs to happen to the histones? Okay. Okay, 5th question. In your CHIP assay, you compare the amount of acetylated histone. So now, again, they're doing a CHIP, right? And remember, CHIP is you're using an you took nuclear lysates, you extracted the nuclear chromatin, the proteins in the DNA, and you sonicate it. You actually bust it up into small bits. And then you're going to use an antibody to grab and pull down a particular protein. This time, they're not doing RNA pull downs. It's a chip assay, and they're pulling down acetylated histones, specifically acetylated histones. And they're measuring that HERC2, so the HERC2 intron. They're measuring by PCR. They measure how much of this DNA here is getting pulled down. Okay, so again, pulling down with acetylated histones. How many acetylated histones are at this position? To determine that, they do PCR of that region. Okay, so HERC2's intron 86 bound amount of acetylated histones. So how much of acetylated histones at HERC2's intron 86 bound to the OACAT2 promoter? Wow, that's a mouthful, right? But anyways, so how much of these at this site are you finding acetylated histones? And so you can see here again the red line and the blue line. So the red bar on the graph represents an intron, this intron here, with what SNP? Remember, so this SNP corresponds to this, and this SNP corresponds to that. Which SNP do you think it is? 5, 4, 3, 2, 1. So 80% of the class, still good, but we're a little bit reduced here in our confidence. 80% of the class chose SNP-T, chose A, SNP-T. Okay, why is that? Yep. I was waiting for you to link it to expression. It's important to link it to expression, right? I know that in the dark pigmented cells, I have more expression of this gene, okay? We know that this element regulates this gene. And so what cell type do I expect to find there to be more acetylated histones at this position? Well, the one that has more expression, right? Because in that cell type, those you would expect the histones to not be, they'd be acetylated. They're not going to be holding on to that particular region to allow access to RNA polymerase. So RNA polymerase could express, could transcribe the transcription factors, bind, and allow the transcription of the OCA2 gene. Very good. All right. So this is what the two SNPs look like. Essentially, this is if you're not kind of having a hard time picturing it, this will help you picture. So we got the SNP-T here. This one has high levels of expression of OCA2. And we have the other SNP over here with low levels of expression. And so, the histones at that particular location are acetylated. They're more acetylated. That allows the transcription factors to come in and do what? The transcription factors don't do transcription. The polymerase does, right? What does the transcription factors do? They're going to hug the DNA at that particular position. They're going to bind the DNA, yes. And so they can, right? So more acetylated histones. The histones don't have the DNA locked down. They're not keeping it all for themselves. Transcription factors are allowed now to bind. And then they're going to activate the expression of OCA2. So they show you that actually, if we were to map it out, it has a conformation like this. Those transcription factors are bound, bringing that element close to the proteins, close to this transcription start site. And just having this conformation, they've omitted a whole bunch of proteins, but you can imagine right where I have my cursor here, proteins being situated there that help the polymerase land on the promoter and do iterative rounds of transcription over and over again. And they facilitate the binding to the promoter. Now, in this case here, they're less acetylated. Histones are grabbing the DNA more tightly. Transcription factors can't bind. Transcription factors are not helping with the expression of the gene. Okay, great. Where would you expect to see more acetylated histones in OCA2 enhancer regions? Like what cell type? Is this a trick question? Where would you expect to see more acetylated histones in OCA2 enhancer region? We're talking about the same OCA2 enhancer region in two different cell types. Same DNA, different cell types. Five, 4, 3, 2, 1. Okay, so, 89% of the class say in melanocytes. Why is that? Why? Yes. Yeah. It's a good get. Like that's right. That's what you and I hypothesize, right? It's in melanocytes that gene OCA2 is involved in making pigments, specifically in that cell type. Liver cells don't make pigments, not that I know. So we would expect the histones to be acetylated that regulate that region that regulates the OCA2 to be acetylated, particularly in those cells. Okay. Now in the liver cells, why not in the liver cells? Because we don't really expect the liver cells to express the genes that make the pigmentation, right? Like the liver cells, they're not pigmented. You have the eye cells that are pigmented. We said that. And the skin cells, we didn't say anything about liver cells. Okay, so why not acetylated? Because in that cell type, we don't really need to express that gene. So if we had to make a hypothesis, we would hypothesize that they're not acetylated. That enhancer region is not acetylated in that cell type. Cool, good. All right, now this is a different example here. Does everyone, before I move on, does everyone get that? Different cell type, identical DNA, one cell type expresses a gene, you'd expect it to have acetylated histones at that particular gene's regulatory site, so it can allow expression. And the other cell type, identical DNA, doesn't get expressed, that gene doesn't get expressed. You wouldn't expect to have acetylated histones at the site, because if you did, you'd probably have expression. So I guess the conceptual leap here is, and it's sometimes a little bit difficult to accept, but at sites where you find acetylated histones, enhancer elements are usually active. They're accessible, okay? The gene that they regulate is expressed. Okay, in this example here, so it's a different example. The product of the agouti gene, is it a agouti or agouti? Nobody cares. Okay. The agouti gene is, the product of the agouti gene is a signaling peptide, the agouti signaling peptide. Oh, that's interesting. Okay. In mice, when the agouti gene is unmethylated, the result is the agouti phenotype, okay? Characterized by yellow fur, the beige fur here. Compared to the brown fur normally found in the strain of mice. What levels of the agouti signaling peptide do you expect in agouti mice versus brown mice? So we're expecting here for you to look at this point right here, unmethylated. When the gene is unmethylated, the result is the agouti phenotype, unmethylated. Somebody's putting E. There is no E. No D either. Come on, man. Yeah, right. Okay, 5, 4, 3, 2, 1. All right. 65% of the class say C, 34% say, C. B, say B, 65% say B, and 34% say A. So majority of you are saying B. What levels of ACIP do you expect to find in the agouti mice versus the brown mice? You're saying higher levels of ACIP in the agouti mice. Why? Please, someone else. I like your guys' answers. I just want to get more class involvement here. Yes. Thank you for raising your friend's hand. That's allowable. Yeah. Okay. Yes. Right. Yeah, contrary to our last example here, in this example, when that gene is expressed, the ACIP gene, right, the agouti gene, it results in the lighter color phenotype, the beige phenotype, right? And so if the gene were methylated, you would expect, what would you expect? More or less expression? Less expression. Methylation of DNA, it generally negatively affects, you guys watch the videos, it generally negatively regulates a gene's expression. It doesn't allow transcription factors and stuff to bind to start the expression of the gene. So by saying that this gene is unmethylated in the agouti phenotype, you'd expect, since it's unmethylated, you would have higher levels of expression rather than when the converse, if it were methylated, you'd find lower levels of expression. Very good. We 65% of the class though, so our certainty, our confidence went down in that one. So yeah, a little more complicated, sure. Okay, so yeah, look at these mice. Wow. Varying levels of the agouti phenotype. Okay, the mice here shown here, believe it or not, they are genetically identical. Okay, they're genetically identical. First of all, is this a quantitative trait or is this a qualitative trait? Yes. It's a qualitative trait. Do you guys believe, is it a qualitative trait? Remember, the qualitative trait is you have a range of phenotypes, right? And if I were to ask all of you to sort these into two extremes, the brown bin and the beige bin, the agouti bin, probably not everybody would do the same same mice in the same bins. It's a good example. This is a qualitative trait. Very good. Yeah. So it's a qualitative trait. And, you know, this agouti, this color here is actually a marker. We call it a marker. We have these in plants and all kinds of organisms. You know, you saw probably in other courses, you have the fruit flies and you got the red eyes or the brown eyes. Like you saw, they got color, right? This is a color marker and it reflects the amount of expression of that agouti signaling peptide. So these guys have really high, these ones really low expression. Now, this was an experiment where the researchers gave the mice they fed it a demethylating agent. Okay, they fed the mother a demethylating agent. Demethylate, what does demethylating mean? Demethylating. Demethylate, yeah. So, yeah. Oh, okay. I think I see where you're going with it. So the demethylating agent is going to remove methyl groups. Remember when we're talking about methylation here, we're actually talking about the DNA gets methylated, not the histones, right? But demethylated, so it'd be removing methyl groups. And what would that do to expression? If you had some thing, a demethylating agent that could remove the methyl groups around the genome, what would you expect to happen to the gene expressions? Increase, right? Right. Okay. So you expect them to increase. So, all right. So, increased methylation of a promoter decreases transcription, right? And so, a demethyl transferase will remove the methyl groups. A demethylating agent, anyways, would remove methyl groups. But you can also have things that encourage methyl groups to be added. And so they get added to the DNA. And when methyl groups are added to the DNA, RNA pol and transcription factors, they generally can't readily bind. They cannot readily bind. Okay, it's only once you remove those methyl groups that all of a sudden you can get the expression of the gene. Okay, so, So one experiment that the researchers did is they actually fed the mice folic acid. And folic acid, ultimately, it does the opposite. It's actually like a methyl donor. It's going to donate methyl groups, okay, and allow methyl transferases to methylate things like the DNA. So when you're fed folate, who of you have had a diet, who have eaten something today that was high in methyl groups? Did anybody eat broccoli? I did. I ate some broccoli today. Broccoli is high in methyl groups, right? So if you have foods, for example, that are high in methyl groups, or folic acid is a good example, what they'll do is they'll increase the methyl content, and that increase in methyl content results in more of your DNA being methylated. Okay, so what they did is they took this folate and they fed it to the agouti mother. Okay, and then that agouti mother had offspring. So what happens when maternal diets are supplemented with methyl donors? What do you think? Is it going to make the color darker, or is it going to make the color lighter? Okay, so let's think back here. We got ACYP, and if ACYP's expressed, we got agouti. Okay, remember we said it's unmethylated, we get expression. If we eat lots of foods with methyl groups, it becomes methylated, and you get less expression, you get less agouti. Okay? So, unsupplemented moms not taking in the folate in their diet, they don't have an abundance of methyl groups, they show the agouti phenotype, high levels of agouti ACYP expression, right? But then they supplement it in the identical moms, they're identical. They supplement the folate into the diet, and look what happens to the color. We have a lot darker color. Why? What happened? They found out it's silencing that expression. It's suppressing the expression of the ACYP gene, the agouti gene. Okay, so got a pregnant mouse. Experiment, supplement. You're going to supplement the diet with folate-rich foods. Now, who are the epigenetic changes happening in? So do you expect them to happen, with respect to our results, the results that we found in the picture, who is actually going to have the changes? Is it going to be the pregnant mouse, or is it going to be the offspring? Five, four, three, two, one. Oh, 68% say B. So most of the class are saying B. Why did you say B? I'll give you an explanation, but why did you say B? Yeah. Very cool. Did everyone hear that? The phenotypic changes that we saw, I specifically stated that they happened in the offspring, right? So if they're happening specifically in the offspring, where would you expect the epigenetic changes to be happening? The offspring. Okay, so I think I want you to step back here and think about this, because we got a mother mouse. It's being fed a high folate diet. We know that folate now we know if you eat a lot of folate, you eat a lot of broccoli or something, you're going to get a lot of methyl donors. Okay. So the mother's eating this, and as the mother eating this, let's say we started to feed it like, I don't know, like 2 months before it got pregnant, and then we kept feeding it throughout pregnancy. Did the mother mouse already have fur at that point? Is the mother going to grow more fur? Okay, so think about the timing and the cellular activities and stuff, right? These, by contrast, the offspring, they're developing embryo, and then ultimately, a zygote embryo, multiple complicated developmental stages. But if they're fed high folate throughout those stages, the gene stays off, remains off, this stage, the next stage. And over time, whenever the cell is really determining, okay, am I going to be a goody color? Am I going to be a brown color? That key point in time, that short window of time, whenever that happens, if you've fed it all throughout its development, you're going to hit that window. Are you going to hit that window in the mother mouse? Well, the mother mouse has already developed. Its cells have already kind of committed what epigenetic state they're going to be in the vast majority of their life cycle. You do have some stem cells that are going to keep producing new cells. Those cells might be affected. You might gradually see the mother becoming darker and darker gradually over time. But the major effect that you see is in the offspring. Okay, because you're providing it at the crucial stage when all those cells are being formed, and whenever the decision to be a goody or brown are being decided. Okay, so the agouti gene is pleiotropic. I like that. I like the big one. It's what does pleiotropic mean? Oh, dude. Yikes. You guys are going to have some hard questions coming up. Yep. Pleiotropic. Do you guys remember? You guys know pleiotropic, right? Pleiotropic. Pleiotropic means a gene that does more than one function. It has multiple functions. Most genes do. This agouti gene, the gene that's, we're talking about the signaling peptide, it's a signaling peptide. Chances are it's going to control multiple things. Not only does it control the hair color, but it also controls the weight. These mice are somewhat, they're a little bit heavier, they're heavier. They have a higher risk of diabetes, a higher risk of cancer. They're even more aggressive. Their behavior is modified by this signaling peptide, okay? So there's really, really wide variation of effects. Okay, so I guess the importance here is to understand that that the changes that we're looking at, in this particular genes expression has multiple effects. But also, if you were to feed a mouse this folate, do you think it's only the agouti gene that's being silenced by increased methylation? It's likely the entire genome, right? The entire genome that's accessible to methyltransferases. Wide variety of effects. Okay, so anyways, how does a DNA methylation, what is DNA methylation may what? Do what? Recruit histone deacetylases. DNA methylation coincides with reduced gene expression. So how does that reduced gene expression happen? Maybe the methylation actually recruits deacetylases, so they remove the acetyl groups from the histones. Maybe they inhibit the binding of transcriptional activators. Maybe they promote the binding of transcriptional repressors. They can do a lot of things, but they generally inhibit the gene's expression. And methylation may be regulated by a transcription factor by recruiting methylation. So you can have a transcription factor that binds the methylated groups and then recruits more methyltransferases or demethylated. Anyways, so it can be a complicated story. Okay, so just at 1 minute, you're looking at the OCA2 enhancer regions in liver. This is really important. Okay. In liver cells and melanocytes, let's say acetyl groups are available in both types of cells. Okay. What else might differ between the two cell types? So in other words, you have these two cell types and the liver cells and the melanocytes. And let's say that earlier we were saying, well, I expected to see in the melanocytes, I'm going to have, more acetylation of the histones at that location, so I have higher expression. But what if you checked both cell types and you actually found the same amount of acetylation? Now, how would you explain the difference between the cell types? How would you explain that one cell type expresses ACYP and the other one doesn't? Or one cell type expresses OCA2 and the other one doesn't? If they have the same acetylation patterns, methylation patterns, you're going to say methylation. Very good. I'm going to say all of those same chromatin modifications, they have the same thing. Why then do we have expression of OCA2 in one cell type and not the other? Yeah. Beautiful. Thank you. Okay, very good. Careful, and yes, very good. The DNA is the same. So that both cells encode the same transcription factor. This is just an example. She said transcription factor. So both cells encode the same transcription factor, let's say. But we said that this particular region had the same post-translational modification. It had the same histone chromatin modifications and same methylation patterns at that OCA2 site. We didn't say anything about anywhere else. So maybe the transcription factor that binds that site is expressed in the melanocytes, okay, and it's not expressed in the other cell type. How would that happen? Well, maybe the melanocytes have a signal that ultimately leads to the expression of this transcription factor that's not found in the other cell type, and so on. There's many, many examples. Maybe the receptor that binds to the signal is present in one cell type and not the other. Okay, so I think you guys, if you're really listening to me carefully, you're probably a big light bulb should be coming on soon because this is the second time I've raised this concept. It's going to come back again and again until all of you guys get it. It's going to show up. It's really important. So don't be shy to ask questions about that. This is just an example of what can happen. So you could have one cell type. It's got a hormone, cortisol, that's present on the outside. That hormone comes in, binds a transcription factor, activates that transcription factor. So this is an actual thing. This glucocorticoid receptor is a part of a protein. We use it in research, but in humans and in animals, it's a real thing. There's a transcription factors that have this glucocorticoid receptor. They don't function. They remain outside the nucleus until that glucocorticoid, that hormone, cortisol, or another steroid hormone, binds the receptor. Then they change conformation and enter the nucleus to do their function. Okay, it's just by the change of conformation. One cell type could be exposed to cortisol. The other cell type might not be. There could be your difference in expression right there, even though you have different methylation or acetylation patterns same methylation patterns at the target. The target might not differ, but the upstream events might. Okay, that's it for today. Remember, deep question 3 gets released tonight. Remember, deep question 2, the feedback is due, I think tonight or tomorrow too. Tomorrow? Tonight. Check that out. Okay. See you guys.